Thanks a lot, Eleni. I would like to thank you, Jota and Tiago, for this very nice meeting. Of course, I feel very much intimidated being the first speaker and having all these nice and famous scientists in the program. I will try to start and try to fulfill the wish of Eleni, starting from the fundamental building blocks of this, from neuron to system and to learning that Eleni described, telling you about something that we consider the basic building block units of neuronal excitability, which is ionic channels. And I will basically tell you a little bit about why it might be important and relevant to model their non-deterministic properties in order to account for channel noise. So the noise, the intrinsic noise that excitable systems, so voltage-gated channels might possess, and why they might be relevant to be reintroduced in modeling neurons at the level of conductances, at the level of ionic conductances. So just for me to know in the audience, are you all familiar with the elementary biophysics of a neuron? What ion channels say? Sodium, potassium, what is the membrane permeability basis of neurons and excitable cells? Please, if you have doubts during my presentation, please stop me and I will try to clarify. So I will not spend a lot of time emphasizing that neurons are somehow electric devices and they are communicated by pulses, and these pulses are indeed the result of some excitable properties, which ultimately refers to ion channels, to how the permeability through the membrane, through the biological membrane of these neurons allows ions to cross this membrane. And basically the information is thought to be encoded in the pulses, in the timing, in the firing rates, and this is not only the way information is represented, it's also the means of information transmission, and you probably are all familiar with the concept of an action potentials. And also, ion channels and excitable properties are important for the neuronal communication. And here in this example, I'll be showing some cortical neurons from the rat visual cortex and their complex morphologies. In this talk is going to be neglected. I will just focus on just zooming on the membrane, something that you don't see here, on how the excitable properties are arising, are emerging. And so this is the picture. On the top left, you actually see a blue neuron. It's a Photoshop. It's not really a blue neuron. It's an image that has been colored. What you normally get with a differential interference contrast microscopy. It's an acute cortical slices, and the structure that you see on the left is a glass pipette. It's a top section of a kind of conical glass pipette, which is normally used for recording and characterizing the electrical activity of these cells. Instead of impaling the neuron, basically making a hole inside the membrane, you actually attach the pipette like it's shown on the right. This technique is known as a patch clamp, whole cell configuration patch clamp, and it has been somehow invented by Nair and Sackman, who were awarded later with the Nobel Prize for discovering this. And I'm just telling you, mentioning this, because this technique allowed to verify some of the intuition of Hodgkin and Huxley, the first to describe quantitatively the ionic basis of excitability. And Nair and Sackman could prove that ionic permeability, so the fact that ions can cross the membrane, and because ions are charged, electrically charged, they will determine the electrical activity of the neuron, is related to microscopic and so discrete and microscopic pores, which can open and close depending on the membrane potential across the membrane. However, these are not only discrete, so it's not the continuous properties of the membrane, they are discrete pores, but they are also extremely tiny, they are microscopic, and as all microscopic systems do, they are sensitive to thermal fluctuations, and by using this electrode, these pipettes, to sense the flow of currents, and this is indicated below for say a few tens of milliseconds or 100 milliseconds, you can see electrically, and this is the reason why Nair and Sackman got the Nobel Prize, you can see the transition, the opening and closing of these channels. And what you realize is that this is not something that switched from the open state to the closed state, or say to the closed state, from the closed state to the open state, but rather is randomly fluctuating between two states. What you actually see is that there is basically almost no relationship with the with time or with the external applied voltage that in this case has been clamped at, probably this is a potassium channel, say clamped at minus 20 millivolt. Instead, you actually see that this is fluctuating and opening, suggesting that the channel is opening and closing spontaneously. And I will try to convince you this is extremely important, particularly along the lines of investigating why non-deterministic properties, why fluctuations, why spontaneous intrinsic noise sources might determine how neurons are responding to signals. And in particular, because this is an area of interest of my group, we are not focusing on other sources of noise in the nervous system, so we are not focusing on the noise, intrinsic noise in sensors that might be, for instance, photoreceptors in the retina, which are by far not perfect sensors. We are not focusing on at the level of when two neurons are communicating and their transmission is unreliable in a way, so it's noisy in this sense, it's a noisy communication channels. We are not focusing on the motor noise, the way actuators are somehow implementing input signals, control signals, resulting in a non-perfect, non-deterministic trajectory, for instance, but instead we focus at the level of ion channels and focusing on this spontaneous transition between closed and open states. So just one first example why you should take into account, whenever you look at the nervous system and you want to quantitatively model, why you should take into account intrinsic noise sources. For instance, the precise spike timing, the emission of an action potential, that is by many thought to carry information. So the precise timing, if a spike occurred now or 1.5 milliseconds later, might have quite important consequences. For instance, affects the precision, and the intrinsic noise is exemplified by these experiments performed in 95 by Meinen and Sejnowski, published in Science. It's a very nice paper, very simple, very short. I would suggest you to read it, in which they did some very simple electrical stimulation through the same pipettes. They were considering slices, so in vitro pyramidal cells from the somatosensory cortex of the rat, and through the pipette they've been injecting a current, and the profile of the current has been indicated in the trace, in the middle trace. So basically they switch on the current, and as you learned from textbook physiology, the neurons started to emit, because the current was sufficiently large to make the voltage exceed some excitability threshold, started to fire regularly as a kind of pacemaker. And however, they were not satisfied doing it once. They did it, say, 25 times. They switch off the stimulus, they waited a few seconds, and then they restarted. And something that may or may not be particularly surprising, and definitely it's not at all replicated by most common mathematical models of action potential generation, the real neurons are not replicating, not reproducing the same timing across all the repetition. This is exemplified in the upper trace, where all the repetitions have been overlapped, and in the lower panel, where just the timing of the spikes has been indicated by a small bar. And you can actually see that the very first action potential, the one that is triggered to the onset of the stimulus, is highly reproducible, but all the others are kind of sloppy. Why this is relevant? This is probably setting a limit of the precision that spikes can generate, and it is worth trying to understand, because this noise generator is by no means constant or uniform or stationary. Instead, it seems to be voltage dependent. As an example, they suggested another protocol in which they did not inject a pulse, they did not switch on the current for, say, 1 second, but instead they've been injecting over and over for, say, 25 repetitions a current whose profile was designed as it's a realization of a stochastic process, but it's just the current that is fluctuating. And sometimes it fluctuates towards hyperpolarizing values, in a way kind of resetting the membrane potential. So if you just focus on the red box that I draw there, sometimes not only that you can see clearly that the reliability and the spike precision is much better, so the same neuron can perform better depending on the trajectory of the membrane potential imposed by the input, you can see that the trajectory is becoming is say it's exploring negative or more hyperpolarized membrane potential. And perhaps this is the reason why, and it is the reason why, you have a kind of reset. You silence this intrinsic noise generator. So noise is affecting precision, not in a trivial way. It's not simply some, if engineers are among the audience, it's not just a noise generator that you add to your equation. It's more subtle than that. And different neurons, in this case I've been comparing in a paper published by Bertobacci and Hugenhardt in 2006. It's a neuron paper. The response, it's the same protocol. The experimenter been injecting a constant current, repeating over and over, in a fast spiking cell, so in another cell type of the cortex, of the somatosensory cortex of the rat, and comparing the reliability, the precision, to the same protocol performed in pyramidal cells. And you can actually see that different cell types have different properties. So I think that this has to be absolutely replicated. I will not go into this for the moment, and suggest that the recent interest in building very detailed models, and I'm referring to the Human Brain project that has been funded as a flagship initiative by the European Commission, as well as the DARPA, so similar initiatives in the States, have been relying on deterministic large-scale description of how individual neurons are described. And what you see here is a beautiful 3D fly-through a neuron. It's a cortex of Henry Markram, and the color is depending on the local membrane potential. So this is a kind of 3D multi-compartment model. We will hear more during the day. And each small piece of the simulated membrane is, however, not incorporating, or not commonly incorporating, this intrinsic noise sources. So I'm suggesting that it's absolutely imperative to introduce and to keep into account this feature. Still, we rely on something that has been proposed more than 50 years ago, 60 years ago, by Hodgkin and Huxley for modeling the excitable properties in a deterministic fashion of every single small patch of the membrane, of the neuron, of the model neuron that I showed you before, this color glowing 3D neuron. And particularly in this paper by Hodgkin and Huxley, that is somehow started a field of, or it was one of the papers that started the field of computational neuroscience, the different ionic permeability through a patch of membrane have been described as voltage-gated resistors. So something that is could be modeled by some differential equations. Here I will not enter right now into the details, and these are contributing to set the resistances or the conductances. It's the same, determining how the membrane potential across this membrane patch is evolving in time. What you actually see in the top, it's a charge conservation equation that says that basically the charge cannot be created. It's just if it changes, it's a consequence of current flows, and these flows, for simplicity, have been limited to a set of so this is very generic. It's a set of currents. K is not potassium. It's just K equals 1, 2, 3. So it can be voltage, it can be sodium, it can be potassium, it can be chloride, calcium, etc. There is a term that is not depending on the voltage, it's a leak current, and there might be another term just on the right hand side for simplicity, for convention, might be the one that Mine and Sejnowski have been injecting through the pipette. It's an external current. So these are the reason why you might have a change in time of the membrane potential, just because you are injecting somehow charge or removing charge from the inside of the membrane. And you actually see that the modeling here is assuming that currents are ohmic, so the current is proportional to the voltage. There is a driving force that depends on the specificity of the channel to the specific ionic species, but this is not crucial. And the conductance, so the inverse of this resistance of this path that ions can follow, is not fixed in time, but depends on the voltage according to this first-order differential equation, first-order kinetic schemes. And if you are not entirely familiar, if you want more clarification, please ask me now, or later I will be happy to give you more details. So this is basically, it's the golden standard. It's the basic building block of any single and multi-compartmental model in the field. Models that are detailed at the level of conductances. So that's why in my title it was written conductance-based model. So, as I said, I would like to convince you that it's important to reintroduce the noise and the non-deterministic properties of those conductances. So this modeling of the previous slides is not enough and might be really important to do so, because we want to explore, and this is not entirely known at the level of a network, how these intrinsic noise sources are contributing to emerging states or to computation in a network. Are they detrimental? Are they functional? Are they important for computation? This is not clear at the level of a network. And it might be important to have a mathematical modeling framework to explore this in silico, rather than performing complicated experiments that would not allow you to remove or to control selectively the parameters. Another reason, however, to stay at the level of conductances is because there are many advantages, for instance, the biophysical realism of employing this conductance-based description of neuronal excitability. And also because today, large-scale models are basically becoming increasingly popular, and maybe one day there will be some kind of global community shared facilities where you will upload your simulation, your model, and you will actually get the result of your simulation, and the language that you will have to use, regardless of the programming language or the description language that you will have to use, is likely to be at the level of single ionic conductances. So, how to reintroduce this non-determinism? It might seem trivial, however, it was not considered, it was not a problem considered at the time of Hodgkin and Huxley. It was already a big achievement for them to look at the ionic basis of excitability. So there are basically two ways, and I will go through them, particularly emphasizing the last. That is simpler, it's faster to simulate, and thereby it's probably more interesting for this time of large-scale simulations. The first one is called Markovian, and it relates to Markov models, and I will tell you what a Markov model is. It's microscopic in the sense that every single channel is modeled explicitly in the memory of a computer. So its states, whether it's closed or opening, whether it's fluctuating, is explicitly described. So in a way, it's an exact description, exact at the level of a phenomenological description. By no means it's a molecular dynamics of the pore, of the way voltage is distributed within the pore. No, it's basically saying it's a black box approach. The channel is closed, and maybe a moment later it's going to open, fluctuating, according to some realistic description. The alternative is called Langevin formulation. So it's roughly related to the Langevin equations in physics, where it's a kind of approximation. A molecule in a solvent is constantly hitting because of thermal agitation with water molecules, and it may not be practical, or may not be always useful to model every single water dipole hitting with your molecule of your interest. Instead, you may want to sum all these random hits and interactions by some kind of noise, some kind of stochastic process, some kind of random phenomenon that is only describing interaction in a statistical sense. So it's effective in the sense that it's not microscopic, and it's an approximation, and it might fail. So it is clear that the hypothesis underlying the second formulation has to be spelled out clearly. So looking at the Markovian formulation, as I said, it's a black box approach. So what you actually see here on the right, in a way, it's a phenomenological model. Basically, an ion channel can be open or closed, and maybe can be in intermediate states, in a number of states. This is interesting. It can be experimentally identified if you actually need definitely two states. In a case of a voltage-gated or ligand-gated channels, it definitely can be closed or open. But the channel, in the most general case, could be in several states. And you see here there is no details on how voltage sensors or particular morphological conformational configurations are changed or are described. It just lumps in one of those circles. Open states, or all the others that right now they don't have names. And you have from one state to the next an arrow, like in a graph. It's a oriented graph, and those letters A, B, G, I, F are indicating the probability of making a transition. So to tell you more in details, the unitary time, so the probability per unit time of making a transition, or the rate. those of you who are familiar with chemical reactions would actually see basically chemical reaction. The species A is getting transformed into the species B by some kind of rate. And this rate is actually a probability per unit of time. So what you do in this Markovian formulation, you associate to one state, say a variable that is a binary variable. It can be 1 or it can be 0, just for simplicity. And you actually say, and you can calculate, I will guide you through a very simple Markov scheme in a moment, that to occupy one state, the open state, so when that random variable s takes the value 1, takes a probability p0, and I will po, the probability of occupancy of the open state. However, you don't have just one channel, you have multiple channels, and the hypothesis is that they are independent. They are identical, independent, and so literally in the memory of the computer, you are going to simulate or to keep into account, to keep track of multiple versions of this channel. So you might have one variable like the s for each channel. So say that you have, you know that in the membrane you have sodium channels, voltage-gated sodium channels, and you have, I don't know, 2,000 of these sodium channels. They are all identical, they are likely to be independent, and you explicitly model each of them. You track the state of each of them. There might be speed up and other techniques, but the concept, the core concept is that you keep track of each of them. And what you describe, and this was roughly what I indicated before in the slides where the Hodgkin-Huxley model was mentioned, you have the number, the fraction of channels in the open states which are contributing to the permeability. And in this case, it's not at all given by an ordinary differential equation, it's just given as the sum, by definition, the sum of those variables that are 1, so just how many of those channels are in the open state divided by the total number. So nothing particularly difficult to understand. And once more, you plug this in the same conductance description as before. Over there, the n of t is just the number of channels for every time, number of channels in the open state. And once more, it's a ohmic description, and what you see, V minus E is the driving force due to the specific ionic species the ion channel is permeable to. So there are, as I said, many advantages of following this route, particularly because it's exact, so it's extremely realistic, and fluctuations are captured in a realistic way. You can consider that if experimented, you can measure those probability of transitions, then you are in. The only unknown that you might have is how many channels do I have, how many sodium channels, how many potassium channels do I have in a small compartment of a complex morphology of a cell. And indeed, it can be performing as a basic building block, like what I tried to suggest. You have the current, you have an existing multi-compartment model, and you plug, instead of your deterministic description, you plug the stochastic description. There are disadvantages. It's extremely slow. Maybe you can think that to track the state of 1,000 or 1 million of channels may require not only a lot of memory, but a lot of time to constantly go through and update. Is this channel in the same state? Yes, no. Is the transition occurring? Yes, no. And once more, if you're interested, I can give you details. It's not particularly difficult how to model, how to simulate these spontaneous transitions. And thereby, because the simulation times definitely increases with the number of channels, it might be inappropriate for modeling, for simulating large networks. That was our motivation, to make a tool, to make an approximation that could be easy and useful for introducing intrinsic noise in large networks. So here it comes the second approach, which is, however, an approximation, but you cannot have everything in life, so we have to spell precisely the hypothesis, and we will come later in a moment. And as I said, like in the Langevin equation in physics, you don't look at individual hits and collisions between a molecule and the solvent and the dipoles, the water dipoles, say, in an aqueous solvent, but you try to capture statistically in an equivalent way how these interactions are occurring. So you basically roughly take the differential equations of the Hodgkin-Huxley model or any multi-compartmental or single-compartmental conductance-based models, and somewhere you plug a noise source. Where? If the question is where and how, which are the properties of this noise? It might be worth to consider that it should perform like the golden standard, the Markov formulation, the exact description. So there are many approaches, and I will briefly review all of them. The simplest one is to consider that you have some extra term to the charge balance equation. This has been indicated in red, and that's a stochastic process, say a white noise or a colored noise. If you ask me, does it have to be white or colored, and what is the variance of this noise, how it has to change, is it a constant, is it a function of the voltage, I simply don't know, because it's very difficult to formulate some kind of reduction from the Markov model down to some additive noise that you plug in the charge balance equation. So in short, this is heuristic. It might be relatively fast and easy. It may not be accurate. And as I said, there is no methods that allow you to design precisely. Oh, there are a lot of sodium channels but very little potassium. Oh, then I have to change that intrinsic noise. I don't know. I cannot tell you a recipe how to shape, how to design that noise intensity, or how it depends on the voltage. There is another possibility, which is to plug the noise, to plug this noisy term into the other equation that I showed you for, when I told you about the GIUGLIANO model, which is in this ordinary differential equation, this kinetic first order differential equation that describes deterministically how the population of ion channels change states from the closed to the open state as a population. Well, you actually add some noise over there, and this is referred in the literature as a subunit noise. It's limited to the formulation in which you might have these subunits, these state variables. For those of you who are roughly familiar with the GIUGLIANO model, and you know what the M gate, the H gate, or the N gates are, these kind of things, this might be related only when you can split the dynamics of the fraction of channels in the open state as a product of state variables. And it may not reduce to deterministic approximation, in the deterministic case, when you take the number of channels to infinity, and you want the system to be, and the formulation to be accurate, that when you make an infinite number of channels, then the behavior of the system approximates the deterministic one. Like in a chorus, if we start singing, we are limited, we are 40, 60 people, I don't remember, we are 40. If we were 40,000, individual fluctuation, individual inability to sing, like myself, I will not be able to sing, would actually be washed out. It will not count a lot. And people outside would hear a perfect deterministic chorus. This is the same thing. And what I notice is that basically this approach has been followed in a kind of heuristic way, and it is pioneered by Fox in 2 papers that give rise to a lot of attempts. So this, however, is not accurate. And we spell out the assumptions of our approximation, which are relatively reasonable. You have identical and independent channels, so thereby we are not considering the more recent suggestions that under some conditions maybe there might be cooperativity between channels. So channels are independent, they are not experiencing, they are not changing their states depending on what their neighbors are doing. They all experience the same membrane potential, and they are in the same compartment, and they have the same statistical properties. Their single channel kinetics can be described phenomenologically by a Markov process, which I briefly told you about. So most of the ionic channels are described in that language. There are alternative descriptions like fractal descriptions in which you don't have a finite number of states, but you might have a continuous number of states. It doesn't look like you really need that level of complexity. You might have, and this is not a crucial hypothesis, you just have one state in which the channel is open. You might not have intermediate states, intermediate conductance states, where the channel is partly open, but this indeed is not crucial. And the number of channels is known and relatively large, and large in the sense of one theorem that basically says that you can take the binomial distribution, the binomial profile, the binomial distribution profile, and approximate it with a Gaussian distribution. And if some of you studied probability theory, this is one of the first thing why, or one motivation why you have the Gaussian distribution. It's an approximation for a binomial binomial random variable. And finally, and this is crucial, limiting the applicability only to few, let's say, not to all the ionic channels existing. The biophysical variables, being the voltage, being, say, intracellular calcium concentration or the concentration of a neurotransmitter, have to change slowly in time compared to the kinetics of the channel. So the gating variables have to change in time slowly compared to how fast the channel is changing state. So indeed, we called it in a different way because it's still a Gaussian noise and it's a conductance noise. And we can show, I will not bother you with all the details, that you can actually make a rigorous derivation from the Markov scheme, from the Markov formulation. So that's exactly, it's microscopic, you can actually derive the statistical properties of that. And you can plug this noise, not as an additive term, it's a multiplicative noise, and you actually plug it as an additive term to the normal, conventional, deterministic description of ionic conductance. So in short, and I will not emphasize it too much, if you have an existing deterministic model, if you have a human brain project model of the human cortex, all the neurons are described in full details, all the compartments are basically tracking the individual currents by the same kind of equation, roughly like in the Auction-Huxley model. Well, I can give you the recipe how to turn this model into its non-deterministic variant, which is accurate, which is it's an approximation, but it's accurate. So the idea is that you look at the Markov models theoretically, and this is just requiring probability theory tools, basic calculation of the mean and the covariance, and so you look analytically at those properties of the Markov of the exact models, and you try to use a continuous, so just a noise, a fluctuating, a continuous diffusion stochastic process with the same properties to plug as a conductance noise over there. And in this particular case, it's a combination of Ornstein-Uhlenbeck processes, for those of you who know what these guys are. So it's a colored, it's not a white, it's a colored noise, and you might have to plug multiples of them in an additive fashion, so relatively fast and simple. So let me guide you, just show you very briefly one example before showing you that it actually works and it's, the results are comparable between the microscopic exact and this approximated version. Let me consider the two-stage channel, so very simple kinetic scheme. It's a very simple Markov description. Here I show you how one channel is behaving, but of course I'm thinking of having, say, 1,000 of these channels in parallel. It can be in a closed or in the open state, so and only in the open state the channel is contributing to the transmembrane current. And obviously the overall conductance from this population of channels is related to the individual random variables that can be 0, 1 associated to each microscopic channel, those S. S can be 1 if the channel I is open and 0 otherwise, and resulting in the fraction of channels in the open state. And once more, I told you in the previous slides, you have a kind of probability of occupancy of the open state. And you might have a differential equation that is exactly equivalent to that kinetic scheme. And once more later, or if you're interested, I can provide you the details for this, which tells you how the probability of occupancy evolves in time. And if those alpha and beta, the probability transitions, are voltage dependent, you're going to have a linear or a system of linear differential equation with coefficients that will depend in time by the voltage or by other gating variables, other ligand concentration or calcium concentration, for instance. And something that maybe some of you would recognize is that differential equation for the probability looks very much like the normal differential equation for the deterministic case, for the fraction of channels in the open state. And this is by no means a big surprise. It's because there is a relationship between, in the large number approximation between individual microscopic descriptions and macroscopic description, like in the Auction-Huxley model. So after that, you have everything, and you can calculate the mean, the variance, and the covariance, or the correlation length, and the autocorrelation function of the probability of occupancy as a function of time, or of the fraction of channels in the open state, this NO of T. And I will not go into the details. These are expressions, and the most important expression is in between is the covariance, that looks very much like a symmetric exponential decay that reminded us that we have a diffusion process that is the Ornstein-Uhlenbeck process that can be simulated very fast, so you don't have to simulate 1,000 channels, you just simulate one equation that has the same properties, or can be adapted to match the same properties, the same mean and infinity, the same variance, sigma squared, that is indicated here, and the same autocorrelation time length, 1 divided by alpha plus beta. And what you do, you actually take this process, which can be easily generated by generating a pseudo-random number, C of t, iteratively. This is a continuous equation, however, and you are able to plug this new term, this new noise, and you are guaranteed that the resulting conductance have the same mean, variance, and autocorrelation time length. Let me show you that this is actually working, skipping this. And I emphasize once more, this might be interesting as a plug-and-play approach to existing models. So let me, to conclude, look at the model validation. So, okay, it's interesting, it might be stimulating, it might be simple, involving only elementary concept from probability theory, but does it work? If I compare it with a golden standard that is the Markov formulation, Markov description. And I will look at 3 indicators. The first one, I will look at the statistics at the steady state. So I will simulate a voltage clamp experiment in which this patch of membrane is clamped. It's the transmembrane voltage is kept fixed, and it's the experimenter or the one who runs the simulation that controls it. Or in a kind of current clamp experiments, looking at the properties involving excitable behavior, the so-called superthreshold dynamical properties, or the properties of the membrane potential below the excitability threshold. I would like to test whether this approach is actually capturing these properties. It may fail at some point, but at least I would like it to match accurately those 3 properties. And obviously, because I mentioned that there was indeed a previous attempt that led to many papers, we would like to make a direct comparison that unfortunately was not in the original papers, and nobody really cared about looking into this, with the original method presented by Fox. So I will show you the golden standard, the microscopic exact model predictions. I will show you our approximation, this Langevin. I will show you this subunit noise that has been used so far in the literature, and I will show you that this one is actually not accurate, is wrong. So this is the steady state properties. It's a voltage clamp simulated experiment. On the left, you actually see a 100 milliseconds realization of the dependency in time, the number of channels in the open state. Particularly in the top panels is the sodium. It's a fraction of sodium channels in the open state. I don't remember how many channels were here, but it's probably around 1,000. They are not that many. And in the lower panel, those are potassium, so the fraction of channels of potassium channels in the open state. And what you see in black is the result of a simulation with the Markov models. So the exact, the microscopic, so the truth, the golden standard that we have. And in blue is the actual same description, but with a Langevin approach, the so-called subunit approach by Fox. And you see that the sodium fluctuations spontaneous fluctuations are underestimated, whereas the potassium fluctuation are overestimated. And what you actually see here is simply the distribution of the amplitudes. You see that the mean is okay, although there are problems with the mean when n goes to infinity, and the variance is actually wrong. You see that the black and blue plots are not coinciding. And something that might be relevant, we don't know exactly whether this is relevant, the speed of this fluctuation, so the color of this fluctuation, or the autocorrelation function, is completely off comparing sodium and potassium induced fluctuations. Instead, because we derive it analytically, so this is not surprising, we derive analytically the properties and we created a stochastic process that matched them, you actually see that for potassium and sodium, I apologize that they are swapped, you have a good match with the mean and with the variance and with the autocorrelation length. Let me go, this is trivial because you say, well, okay, but it's you plugging the mean, plugging the variance, so it's not a big surprise that when you fire up the model, you actually get a good, accurate approximation. What it might be more important is to look what happens when you have sodium and potassium channel populations all together in a single compartmental model. And we took the Hodgkin-Huxley model that's right now, so it's a single compartmental model
composed by voltage-gated sodium and voltage-gated potassium channels. And what we have now, it's something that you normally do not have. It's a model that is non-deterministic in the sense that, say that you apply a very small current pulse, a deterministic model would actually behave very boringly, always the same, firing if the current is sufficiently large, or always responding as a passive RC circuit if the current amplitude is not large enough. Instead, what you have here is that sometimes you can actually elicit an action potential, like in a real experiment, but sometimes, I think it was 2 out of 10 times, or roughly, I don't know, 8 trials that we attempted, sometimes you might have a failure, which actually to me is intriguing, is interesting, because it tells you that indeed you have the same box, and these intrinsic noise sources can determine whether or not the neuron is going to respond. So for the dynamical properties, we will look at whether or not you have a failure, so how many times you actually have or not a response, that we call efficacy. Sorry. We will look when the action potential is emitted, which is the latency. You might notice that the latency is not always the same, it's just fluctuating, and we will also look at the jitter, so the standard deviation of these latencies, not only the mean but the standard deviation of the peak of the elicited action potential. And once more, we indicated, we studied 3 cases: the microscopic, by exact, by Markov models, which is in black; our description, which is based on the Langevin equation, the Langevin concept that we called effective, in red; and the current method, the subunit noise, proposed by Fox. And on all the panels, what we did, we've been increasing the amplitude of the currents that we've been injecting, and looking at how many times you have a spike or you have a failure. And of course, this count is between 0 and 1. At some point, if the current is strong enough, you are going to have 100% of the time a pulse. You're going to have a decreasing latency the stronger you push the neuron. Indeed, similar to what we observe, for instance, in slices or in dissociated cell culture. And you also have a decrease in the jitter, the stronger is the input. And you can see that in these 3 indicators, or also in the distribution of the latencies, so not only the mean and the variance, but just the distribution, we actually have a very good match between Markov formulation and the approximated Langevin equation. So in short, if you want to turn your model into a non-deterministic version, please use our method. It's fast, it's convenient, and particularly it's accurate. Do not use Fox method, which seems to be off. We look at some other standard feature that you can approach experimentally, which is the so-called frequency versus current curve. It's a kind of input-output response investigation. The stronger you stimulate the neuron with a DC constant amplitude pulse, the more spikes you have in the Hodgkin-Huxley model. And here we do not show the Fox model because it's completely off. We actually have an intermediate region in which you might have intermittency because of this non-deterministic properties, and our effective approximation is actually nicely behaving like the Markov microscopic description, but without the heavy computational load. So it's okay, it's nice for the active properties. And what we also look at, we look at the subthreshold fluctuations, and we study the power spectrum. Let me go to the power spectrum first. The power spectrum of the subthreshold fluctuations, and once more comparing Markov with the Langevin thick lines versus thin lines, we have basically roughly a very good accuracy and some voltage dependence. That is what we wanted to achieve. So particularly if you clamp the voltage, if you inject the current, slightly depolarize the membrane, you start having that this fluctuation might carry some resonant information, so around 2 Hz. This is known, and experimentally you actually see the same. Let me conclude looking at the initial motivation for replicating, for instance, the unreliability of neurons to replicate the same spike timing across several repetitions. This is the experiment that I showed you. So we could perform, and I will not show you on the same slide because by no means we wanted to match the same firing frequency or the same latencies. We took the Hodgkin-Huxley model, simulating it by the Markov schemes or simulated by the Langevin schemes, and we compare it once more, black versus red, and you see from the raster plot every small dot is the firing of an action potential as a function of time, or as an histogram of all these events. There is a good match of the lack of precision of the neuron in repeating itself, both when the input current is a constant DC current, as well as when the input current is strongly fluctuating, thereby resetting this intrinsic noise generator. You see that on the left, the histograms are, so these curves are relatively large. Instead, here they are more compact, indeed, because the jitter is lower. So let me conclude. I show you, and with this excuse of showing you this procedure, I hope that I introduce you with some language or some tools of conductance-based modeling. I focus on single compartments, but I hope that you can think that how easy this is to be generalized in simulation of multi-compartmental models, particularly focusing on one aspect, this intrinsic noise sources, channel noise, so-called, and show you that it is possible by simple probability theory. I did not have time to go through it in the details, to derive an approximation which is precise in the sense that I described, that it's matching the golden standard predictions, is fast because it does not depend on how many channels you have in your system. It's actually one additional equation to the system, to your existing set of differential equations. Particularly, it's a bit faster than the fastest macroscope. Sorry, it's faster than the microscopic algorithm, the fastest Markov algorithm, and it's a little bit slower than the subunit Gaussian noise method. It's general in the sense that this can be applied to any voltage or ligand-gated channels, provided that it satisfies all those hypotheses. And this is our idea. This can be finally used to test consequence of intrinsic noise, channel noise at the level of the network. This is very difficult to do it experimentally, and you might want to do it in silico. And it's also interesting for us because this would allow for some very fancy experiment to plug back or to try to alter or to try to play with in a real neurons with a channel noise, intrinsic channel noise sources by some computer-controlled dynamic clamp protocol in which the experimenter is injecting something that depends in real time on the membrane voltage of the cell. And I show you that it actually fit nicely, replicate nicely steady state and active properties. Let me conclude acknowledging funding and particularly Dr. Daniele Dinaro, postdoc in the lab, who actually basically did most of the work that I presented today, and I thank you for your attention.